Thank you very much, and thank you all for being here this morning, and special thanks to Ivan and Massimiliano for the invitation. And I was revising my slides left and right yesterday during the many discussions that I heard after talks. So given that there were not that many information theorists present in the audience yesterday, I thought it would be good to start with a very brief review of what Shannon's information theory has done so far and what its limitations are, not just in biology, but for many other applications. And then shamelessly advertise an NSF-funded center, the Center of Science of Information, that has been going 11 years now, and that has really tried to come up with the Shannon 2.0 version of his theory, which may address some of the questions that are very relevant for biology. In the second part of the talk, I will go ahead and talk about my own research and mostly about coding theory, and in that context, coding for synthetic biology. So what is information and coding theory? For us that worked in this field for many decades, I would say, it's been a very pleasurable and good experience. Two gentlemen depicted here, Shannon on the right, and one person that is impossible to leave out, but unfortunately he's usually left out in the discussions of information theory, is Kolmogorov. Those two are really the ones that gave rise to modern probability theory and information theory. And the reason why they're intrinsically coupled together is that information theory relies really intrinsically on probability theory and probabilistic arguments. So Shannon in particular was interested in one problem, the problem of data transmission or data storage. And I don't know if this picture can be seen clearly, but in his original framework, in a number of papers he discussed, the setup is very formal. You have a source, which is described in terms of a probability distribution. You have a system that we call source encoder, and then you have a channel encoder. And I'll go into details in a second regarding what these concepts mean. Then there is an optional part that is used in some communication and storage schemes, not used in others, which involves modulators and demodulators. And then everything you do on the upper side, you have to undo. And as I mentioned, the two components that we have, or the multiple components that we have here that constitute an information theoretic framework, information theoretic framework really rely on concepts such as information theory that is inherently probabilistic and, not to be forgotten, asymptotic. Usually information theory tells you what is the best you can do with the system, while coding theory, which appears here in the source encoder and channel decoder, is on a more practical construct, and it's mostly deterministic. There have been many probabilistic coding constructions, but they don't tend to be implemented in practice. And it operates in the discrete domain, mostly again. There have been codes that are really focusing on analog signals, such as rank modulation codes and other codes, but codes are really mostly deterministic, and they operate in a discrete domain. So what is a source and what is a source code or source encoder? A source is anything that emits symbols according to some distribution or generates waveforms according to some PDF. So once you have a source, and I don't know why this hand is floating around. Once you have a source, the first thing you would like to do is remove redundancy from the source. And the reason why you want to remove redundancy from the source is a lot of redundancy is hurtful in the transmission process because it slows you down. Same thing for storage, because it increases the storage footprint. What source coding really does is it removes redundancy in such a way that you can still reconstruct the original signal back if it was discrete, or it performs some form of quantization. And a lot of you must have heard about quantization, which is a lossy compression method, which allows you to go from an analog to a digital domain. On the other hand, this channel encoder is something that is designed to address the behavior of the medium through which you're trying to transmit or store information. What that means is basically a channel coder is trying to address the errors that will occur in a channel. And to be able to do channel coding, you really need to know what your channel is. You can abstract it, you can formalize it in a way that is very simple, but it has to be clearly stated. And the modulator blocks are basically something, as I mentioned, that is optional. It relies on the fact that you know beforehand some characteristics of the channel. They're not random. Those characteristics you know, and you want to adjust your signal to the known characteristics. Channel coders really try to address random errors. Constrained or modulation codes try to address errors that you know beforehand would occur. And so cryptography is optional. It was addressed in Shannon's work, and it had a lot of success stories. So every recording system that you can think of, disks, tapes, every wireless communication system, every cell phone, deep space communication, everything uses Shannon's information theory and coding in particular. So these were the big success stories of coding, the good parts. An even better success story, given the craze we are witnessing with respect to machine learning and statistics, is that information theory has an inherently strong connection with statistics and machine learning. So here is a famous book by Kullback. Those of you that worked in information theory or statistics definitely know about Kullback-Leibler divergence. And Kullback published a very influential book on information theory and statistics. And there are so many numerous applications of this area that you can immediately think of in biology, including one area that is very close to my heart. It's distribution and support estimation. It's one topic that has been brought up in biology many, many times in the past. You want to estimate the number of species. And you clearly cannot see every specimen, so you will have to collect a certain number of specimens, say butterflies, and based on the limited number of observations, try to estimate what is really the true diversity in the world of butterflies. What is the total number or total size of the population? One very interesting project that may be interesting to those working in immunology is at some point, Alon Orlitsky, who is one of the pioneers in this field, and I went to the CDC to try to help them predict the viruses that are going to be the most dangerous ones to put in a cocktail for vaccination, for a flu vaccine. And the interesting observation they had is basically we are sampling, we are getting DNA, we are getting sequences describing these viruses, but we really have no clue how many viruses are out there, how large is the population of one type of virus. So if we knew somehow to very accurately estimate the distribution and the support size, we could do much better in terms of designing vaccines. So we did some interesting work along these lines in terms of trying to determine the population of the microbial gut, or the microbial microbiome, basically sampling everything that's in the American Gut Microbiome website, housed by University of California, San Diego. But if you think of problems like that, think of information theory. Anomaly detection, what is typical, what is atypical behavior, that's another thing that you probably don't associate with information theory, but it has been studied in our framework, and it's really something that we can help with. Learning higher order interactions, for those of you that are interested in biological networks, you know that there are a lot of motif structures, and learning and analyzing these motif structures is also a problem that has a lot of connections with information theory. And ordinal data analysis, there were quite a few groups that have been using ordinal data analysis and its connection to information theory to do gene prioritization and address other problems. So this was the advertisement and the praise that I have to give to information theory. But let's step back a little bit and let's face the truth that information theory is not a panacea. It's not going to cure or solve all your problems. What Shannon did not do, and I apologize beforehand, but he really didn't address these questions, so it may be a point of contention. But he did not talk about representation and semantics. He was really interested in communication systems, despite the fact that many of you probably know that his PhD was in biology. But he was really interested in communication systems. So he worked with data signals of very particular types, strings or waveforms. So he wasn't really addressing questions of representation and semantics, context of information and timing. shapes and networks were kind of overlooked in that line of work, later on picked up. And so what is really lacking in Shannon's theory is a unified approach to all these concepts, and on top of it, a unified approach that would apply in the finite regime. Because I mentioned you have to be very careful. Everything that Shannon did was asymptotic, meaning you can guarantee that good things will happen to you if you allow long block lengths or long communication times or whatever you like to call it, and the probability of error in the worst case is guaranteed to go down to 0. But in the meantime, these questions were really not properly addressed. So just the brain teaser, because a lot of you here are interested in puzzle, here's a formula that everyone who has worked even a little bit with information theory will recognize. It's the classical formula for the entropy of a discrete source with probability mass function dictated by these numbers pi. And here is a question that I want you to address, and I'm not even going to bother to solve it. I am not even telling you that it's something that can be easily addressed. So if you use your classical idea of what information is, and then you immediately jump into using the notion of entropy to estimate the average information content or whatever you want to call it, then you will be faced with this question of how do I deal with these 5 words that clearly, if we all agree, carry the same amount of information, at least the fuzzy way we understand information. If you're going to look at different languages, different usages of, or different representations for the same things we want to say, then, and you use an approach, naive approach of just trying to estimate the entropy of these sources, you will see that the distributions are very different. And I didn't even bother putting Arabic or some of the Chinese dialects in here because I wouldn't know how to use the symbols. But you can immediately tell that entropy is just a number. If you don't associate a meaning to it, and unless you interpret it properly, which Shannon did, as basically by saying that entropy is a lower bound on the compressibility rate of a source, then it has really nothing to convey to you that is useful. And that's just one example that we usually put up when we advertise what I shamelessly mentioned at the beginning, our famous Center for the Science of Information. So this is an NSF Science and Technology Center that has been funded for 11-12 years. It's housed at the University of Purdue. It has a large, pretty much everyone who works and who is who in information theory is part of the center. And if you look at the statement, the post-Shannon challenges, you will clearly see this is something we always present when we talk about the center, is that we don't know enough about information and information theory of structures, networks, regulatory networks, protein interaction networks, social networks for that matter. We are really tapping in the dark with the large number of problems related to time and space. And this is really selected by our leading information theorists as a statement saying information theory is at weakest in dealing with problems of delay. So, and I can give you more examples afterwards, semantics and learnable information. So my suggestion would be, if we want to have a great dialogue and if we want to do great things together, biologists and information theorists, it would be very useful to keep in mind that Shannon didn't solve all problems, and that information theorists are still working on a large number of challenges that would really benefit if solved information theory, biology and other disciplines. So all this being said, what I want to talk about now is where and what have people done in terms of using information theory and coding theory in biology in a successful way where you can really not speculate about the accuracy of the results. And this is a biased view because it comes from me, it's my talk. So what I want to say is bioinformatics and computational biology have benefited so much from information theory, probably without knowing it. And again, I'm going back to my favorite crediting of Russian scientists. If you think of your sequence alignment algorithms, and Everyone who ever worked in computational and molecular biology needs to do sequence alignment. Guess what? Sequence alignment is our invention, a coding theory invention. It can be traced back to the work of Levenshtein, who was using it to describe the famous Levenshtein distance, or the edit distance. And I'm very happy to say that the Information Theory Society is acknowledging his many contributions with a special issue in which we managed to secure articles by the likes of Pavel Pesner, Bonnie Berger, Michael Waterman. Thank you, Ubli, for helping with that. So the other thing that Levenshtein did, and it's very exciting, overlooked as well largely, is trace reconstruction. And the words may mean nothing to you right now, but trace reconstruction is a very interesting problem. It says you have a sequence or a piece of information that you're transmitting in parallel or in different time instances, but multiple times. The same piece of information transmitted multiple times. Think of the same signal being transmitted through a bunch of parallel channels. And the goal is to basically reconstruct the signal by looking at the outputs of all these different channels that operate in an independent way and introduce errors, let's say, in an independent way. The question is, how many such observations do you have to have in order to be able to reconstruct the signal with diminishing probability of error, or to be able to basically reconstruct it without any errors? So that's, again, something that came from coding theory, and it has applications in phylogeny. A lot of the analysis or talks I heard yesterday that were mentioning pathways and parallel propagation of signals through pathways may be interested in this topic. Another topic that I think is a success story, it's something that the NIH started maybe 5 years ago. NIH had a bunch of BD2K, big data to knowledge calls for proposals, and one was really concerned with the topic of data compression. You must know that we are drowning in genomic data. A colleague of mine, Jean Robinson, who is obsessing with bees in Urbana, wrote a very influential article, shall we call big data astronomical or biological? Because right now we have so much sequencing data that it's very hard to store, transfer, and exchange it between groups. So the call, the BD2K call, said come up with very, very good solutions for genomic data compression, because our classical Lempel-Ziv approach, classical universal codes, will not work. So one of these grants went to Urbana, to my group, and we did a lot of work on genomic data compression, and the proof is in the pudding. You don't have to worry, am I interpreting it correctly? You get a 20-fold reduction in the size of the file compared to when you use gzip or bzip. You know you're not doing things wrong. If you can run your software, plug in the data, get it back undisturbed, you know it's true. There is no doubt about that. One topic that is very exciting, at least to me, is trying to bring coding theory to synthetic biology. And this is what I'm going to talk throughout the remainder of my presentation. And systems biology, the operational principles of system biology, I wish I could talk more about it, because there is a lot of knowledge that can be gained from network information theory in this concept, in this context, but I really will not have the time to do so. So rather than trying to spread myself thin, I already did by trying to give you a really quick lesson about what information theory can do, has done, and cannot do, let me just focus on this molecular and synthetic biology problem. And in this context, I would like to really gratefully acknowledge a lot of funding from both the NSF, DARPA, or amongst others, DARPA, SRC, Air Force Research Lab, and other organizations. So the talk that follows after this is going to be on a very boldly named concept that I call DNA punch cards. And you heard yesterday from Steve's talk that there has been a hype about trying to store data in DNA. After all, DNA is a very durable storage media. We have nucleotides that can store roughly 2 bits of information, not roughly, exactly 2 bits of information, and the spacing between them is roughly 0.3 nanometers, so you go ahead and calculate the density. We are proud to talk about the fact that We discovered DNA of Neanderthals that are 30,000 or more years old, and we almost managed to get intact copies of a few of their genomes. So it's not a surprise that people started looking into this area. And I think the credit really should go to people from Arizona that did this research and proposed this research much earlier. The problem was they proposed it so early on that no one believed it's going to ever happen. And their work has been largely forgotten, but I would be very happy to point you towards their work. But we usually start by acknowledging Church and Goldman in this context. So you would think this is science fiction, and it really isn't. It's as simple as it gets. You come up with your information, binary, you do whatever you want to do. Do what Shannon tells you. Compress it, protect it with error correction redundancy, and then convert it into a 4-letter alphabet, and then call your favorite synthesis company. My company to go to is IDT, for many reasons that I won't discuss. And then you synthesize your DNA, and voila, here is your data storage system. Clearly, you have to read the content. How do you read it? Again, every lab in the world has, respectable lab in the world has some sequencing device, either Illumina or Oxford Nanopore. And what we did starting from 2015, almost 5 years, is to basically work on enabling random access, protect against various types of errors, enable error-free nanopore sequencing, make the system more portable. And we had really specialized coding techniques that can solve a lot of open questions in this field. And the results that I want to show and the pictures that I want to show is the latest work that we have done in this area. We systematically encoded Marlon Brando images. As you can tell, this is an image from a very bad movie, Nightcomer, I think. The movie is forgettable, but the image was always is a good example to show. And why is it interesting? It's interesting because for the first time, we used no error correction redundancy to encode images and retrieve them without basically many errors. And the idea is to use machine learning. And I pointed out to you that machine learning and information theory are so highly intertwined. This is the original image. This is an image which we got after observing a bunch of errors. And I'm pretty sure you cannot see that his forehead is blue, that there is a lot of purple on his lips. But there is a technique in machine learning called image in painting. And it basically says you have your favorite celebrity, and this is unfortunately the only thing I can find online, not too excited about these, but then you damage the image. You damage the image in such a way that maybe you cannot tell too many details in the face. But deep learning, since we have so many images to deal with, deep learning allows you to train the networks to recognize facial features, and then you can do what is called image in painting and fill out the missing parts. What is interesting about this is the first time that we managed to automatically detect the in-painted or the components in the image that are miscolored or full of errors. And that is done in a way that cannot be replicated in digital technology. We talked to everyone in computer vision. It's a wide open problem. Second, it was done in memory, something that again cannot be done in classical storage systems. So this would be a pattern that shows where do you suspect the errors happened. And then you run an image in painting scheme. You kind of smooth him out a little bit. I'm not so sure that we didn't do too much photoshopping here in memory again of his image. You see the only thing we couldn't really get rid of is a little tiny error that happened here on the lip, the little bruise thing. But the images, and with a little bit more fine tuning, could be easily made to or easily stored in DNA without using any redundancy. And the point of this is that the cost is really what is driving this application. So if we reduce the cost, we don't add any coding redundancy, we're in good shape. So how large was the cost? Going to the elephant in the room, and I know I have very little time left, so I'll try to keep to speed this part up. Let's say gigantic. It's not even close to what the digital technology has to offer to us. So if you look at the amount of money that we spent for, let's say, 8 images of Marlon Brando to get it even without error correction redundancy, it cost us around $20,000. And I know this is where we start choking. and this is not what we want to hear. So the idea that coding theorists, and basically my group and my students who are coding theorists, came up with is, why bother making DNA if you can find it everywhere? Watching forensic files or any crime story, you will see that there's DNA being shed everywhere. You move around, your skin cells are everywhere, your hair follicles are everywhere. So why do we pay so much for synthesis? The problem is I want to store Marlon Brando. As you saw, my agenda is very simple, Marlon Brando. So how do I make DNA encode Marlon Brando? It's not designed to encode Marlon Brando. So information is hardwired. It's in the genetic code, and there's nothing you can do to get this bacteria to store your favorite two subjects, let's say Marlon Brando and a cat. Everybody knows I'm crazy about cats, so it was coming. So how can you store information in native DNA without changing the content? We were toying with this idea. Maybe we can look at the distribution of patterns in certain native DNA strings, let's say the genome of E. coli, and try to figure out what are the smallest number of edits to get our some parts of the sequence to look like Marlon Brando. And clearly, guess what? That's not going to work. So what we saw and what came out as a very interesting idea is that at the time when we were obsessing with our Marlon Brando images, the world of synthetic biology was obsessing with CRISPR-Cas9. Basically, with the big breakthrough and the discovery of this bacterial immune system, which allows you to do targeted sequence editing, basically cutting or nicking DNA strands. Mostly the applications were for native DNA strands. So here is a proposition. Why don't we just take native DNA, which you would agree is almost free? There is some work you have to do and something little to pay to get native DNA. But what if we take that native DNA and try to store information in it, not in the sequence content, which we cannot really easily do because it's not storing what we want to store, but in terms of cutting or nicking, if you have a DNA strand, for those of you that have an information theory background, it's a double-stranded construct. So if I say cut, I mean you cut both strands. If I say nick, I mean that you only cut one of the strands. The DNA still has its structural integrity, but you're only nicking, basically creating a snippet in one of the strands. So can we use these enzymes, something like Cas9, to nick the information into native DNA? And the answer is yes, and this is what we demonstrated in a number of experiments, and it reminded us of an old technology going back from digital storage systems all the way to modern systems that have incredible properties, to the ancient old systems of punch cards. Because if you think of each DNA strand, double helix, as a row in a punch card, you're really going to store your information by punching or nicking the DNA in predefined positions according to the data that you want to store. And since I'm running out of time, I just want to give you a few takeaway lessons from here. So how do I store information? Using statistical analysis, information theoretic arguments, we find locations that we think are very amenable to nicking. We use those locations as the positions in which we are going to store information. Think of the simplest storage system. 0 means no nick, 1 means a nick. And that's great because that's how CDs work. CDs, if you want to store a 1, you burn a hole in the disk. If you want to store a 0, you don't do anything. So you're really doing the work only when you want to store ones. You can do it on both strands, which means you can convey information of log 2, 3 bits. And then you can nick the sense or antisense strand. And you want to be very precise with your nicking, so we use the special enzyme called PFAGO. And we have to place the nicks a little bit apart in order to be able to detect them. And that reduces the density But hey, I mean, reducing the density 50-fold, but saving a lot in terms of the storage capacity is worth doing it. And I will just show you how the whole experiment is done. The beautiful thing is that since we know the genome, the reference genome, or the genome that we're using as a punch card, it is something that is stored even without the need for DNA-based data storage. Everybody has in its library the sequence of certain microbial organisms. We know what the sequence is. And then if these are, let's say, the positions that we can punch or not punch, well, all we have to do is, once we do the punching, if we want to read out the content, we denature the DNA. We get a bunch of fragments, and we align them to the original reference, and we're done. We don't see absolutely any errors. And I want to show you very fittingly, because we're in DC, to show how much we appreciated some presidents in the past and not some in the current. Forget what I said. Here is an experimental readout of an image we took of the Lincoln Memorial. And these are snippets. This was one piece of the image. We just took random blocks of length 10 from this encoding. And these are the fragments you get. These are the fragments that really tell you where the positions of the nicks are. You align them to the reference, and wherever you see that fragment ends and another begins, you know that that was the position where you nicked, that was there where it was a 1. And even if something is missing, if you see a hole, you know that there must have been something in there. Coverage plays very little role. People are so worried about coverage when they sequence data. It doesn't matter when you do it this way. So you see, basically, this is all ones. You see all the 10 fragments, 11 fragments, because you're cutting 10 positions, so you get 11 fragments. And these are the results and the coverage plots that you see for Lincoln's image. So all this being said, I really want to acknowledge, and I'm really sorry I'm over time, a bunch of great collaborators at UAUC, at Arizona State, at University of Texas, Austin, people from all walks of life, chemistry, electrical engineering, the Keck Sequencing Center, then a bunch of people from University of Minnesota, San Diego, Irvine, and our favorite collaborators from EPFL that do such great work with nanopores that help us do the sequencing in a very cost-effective manner. Thank you so much.
That was an impressive arc from the past in the history to the present and well into the future, I think. Questions?
Yeah, I mean, if you're someone from my community listening to your talk, right, keep in mind that we have problems in chemistry that are long-standing, haven't been solved for the longest time. Something very simple to describe, like how do two molecules interact in aqueous solution? We don't understand how to model that. That's why drug discovery is so difficult. No progress has been made on this. We are desperately interested in having people tell us about information, but this is what your comment, and also what Christoph mentioned yesterday, information about what? It's the semantics, it's the downstream stuff that's of interest to us. And so we get struck by the fact that we're always looking for something actionable, but we don't want it to be another parlor trick, should I say this, which is what, in some sense, what George Church storing his book in. It's not something that addresses any big problem. Second, what we do from our community, we look at this and we say, hey, good grief, of all the things that have advanced in civilization over the last 50 years, information, data storage, it's been miracles, right? Whereas the simple question is, how do two molecules associate in aqueous solution? Has seen no progress really whatsoever. So how do you square this circle? How do you speak from a community like ours who see our big problems not being solved, your problem is being solved magically, but no way of getting this information transferred?
No, our problem has not been solved either. I wish, as I said, that information theory can solve everybody's problems. We have a lot of issues with addressing exactly semantics, timing, delay. All of these things are ongoing research. And the biggest issue that I have seen with biology, trying to interpret biology, is there's so many more exceptions than rules. I think that biology really operates at the edge of various phase transitions, as we would agree, and that's what makes everything pretty much non-stationary. And that becomes a big issue for information theory. Stationarity is a key concept that makes a lot of things work. So what we really need, and this is why I really changed my presentation, moved a lot of the results from various areas that I worked in and talked more about information theory in general, is we really need to talk, and we need to have a honest conversation in terms of what we can do and what we cannot do. And my fear is that biology is very complicated. What we can definitely start with is do a better job analyzing biological data. As I mentioned, the proof is in the pudding. When you have the data and someone tells you, this is how many samples you need, information theorists can tell you, to get this kind of error, this is how you estimate a distribution. No offense meant, but there were tons of papers written about estimating entropy. It's not your classical, I count how many times I see a certain symbol, I divide it by the total number of times I saw a symbol, and then I plug that into the formula for entropy. No. Entropy has a term that has a p and a log p, a product. One term is exponentially larger than the other. That creates a lot of problems. Even something as basic as that, it would be common sense, just do the counting, plug it in. That's not how it works. So we need to start with educating the general public of what is the right thing to do with the data. Once we have the data, we analyze it properly, maybe we will understand more about the semantics, about the pathways, about the functionalities in biology, and then we can make some models to hopefully help us get deeper into it. But it's a long and complicated process. I wouldn't lie that it's going to be a piece of cake.
I think we need to transition to the breakouts. So let's thank Olga again.
Thank you.